Welcome to Niga MedGuide. Today we will discuss anatomy and physiology of small intestine. Small intestine is the longest part of the digestive system, extending from the pyloric sphincter of the stomach to the ileocecal junction of the large intestine. Its main function is the digestion and absorption of nutrients. Here is a point that can confuse you is that why is small intestine the longest part of the digestive system? So I want to clear that it is called small not because of its length. In fact, it is longer than large intestine, measuring about approximately 5 meters long. It is called small because of its narrow lumen or small diameter, that is about 2.5 centimeter. Large intestine, although shorter, measuring only 1.5 meters, has large diameter of approximately 6.5 centimeter. That's why it is called large intestine. Now let's talk about the parts of the small intestine. Small intestine has three parts that are duodenum, jejunum and ileum. Duodenum is about 25 centimeter long. It is C-shaped and curved around the head of pancreas. Secretions of the gallbladder and pancreas merge into the hepatopancreatic ampulla. These secretions enter the duodenum at the duodenal papilla. Duodenal papilla is guarded by a ring of smooth muscles, that is sphincter of Oddi or hepatopancreatic sphincter. Jejunum is the middle section of the small intestine and its length is about 2 meters. Ileum is the longest portion of the small intestine and is about 3 meters long, and it ends at the ileocecal valve or junction that control the flow of the content from the ileum to the large intestine and also prevents the backflow. Layers of the small intestine. Like the rest of the GI tract, small intestine has also four layers, and starting from the outside to the inside, these layers are serosa, muscularis externa, submucosa, and mucosa. Serosa is the most outermost layer consisting of connective tissue. It is visceral peritoneal layer that secretes serous fluid that help intestine to move freely in the abdominal cavity without any friction. Mesentery, that is a double fold of peritoneum, support the small intestine, also attaches to the serosa. Next to serosa is the muscularis externa that consists of two layers of smooth muscles that are inner circular and outer longitudinal muscle layer. These muscle layers help in peristaltic movements and segmentations. Next is submucosa, made up of loose connective tissue, and it consists of blood vessels, lymphatic vessels, and submucosal nerve plexus, that is Meissner plexus. The most innermost layer is the mucosa, and the mucosal surface is greatly increased by permanent circular folds that help in mixing of chyme, villi, and microvilli. Let's have a look on the structure of villi. So villi are the finger-like projections of the mucosal surface into the lumen, and they are about 0.5 to 1 millimeter long. Their covering consists of columnar epithelium, enterocytes, and microvilli. Goblet cells that secrete mucus are also found interspread between the enterocytes. These epithelial cells surround the network of capillaries and lymphatic vessels. Lymphatic vessels are called lacteals because absorbed fat give the vessels milky appearance. Intestinal glands are also found beneath the surface between the villi. Small lymph nodes are found on the mucosal surface throughout the length of the small intestine, and these small lymph nodes are called solitary lymphatic follicles. But the collection of 20 to 30 lymph nodes are found in the distal end of the ileum that are called as segregated lymph nodes or Peyer's patches. They consist of defensive cells that provide immunity or neutralizes invading microbes or antigens. Blood supply to the small intestine. Arterial supply is by superior mesenteric artery and venous drainage is by superior mesenteric vein that drains into the portal vein. Now let's talk about the physiology of small intestine. Two types of motility occurs in the small intestine, that are segmentation and peristalsis. Segmentation is rhythmic, regular contraction of small intestine that mixes the chyme with the digestive juices and increase the contact with the villi for the absorption. Peristalsis is wave-like contraction that propel the chyme towards the large intestine. Secretions of the small intestine. Intestinal juice that is produced by intestinal glands and it is enriched with water, mucus and electrolytes. Brunner glands that are located in the duodenum secretes alkaline mucus that acidify the chyme. Goblet cells that are interspread between the enterocytes of the villi secrete mucus that help in lubrication. Brush border enzymes that are secreted at the surface of enterocytes are peptidases, disaccharidases, and lipases. Chemical digestion occurs in the small intestine due to the brush border enzymes. Disaccharidases such as maltase, lactase, and saccharase break down the disaccharides such as maltose, lactose, and saccharose into the monosaccharides. Peptidases breaks down the polypeptides into the peptides and amino acids. Lipases break down the emulsified fats into the fatty acid and glycerol. Absorption of most of the nutrients into the blood occurs in the small intestine, which includes carbohydrates, proteins, fats and fat-soluble vitamins, water-soluble vitamins, minerals, and large quantity of water and electrolytes. Exception is that fat and fat-soluble vitamins are not directly absorbed into the blood in the small intestine. In fact, they are first absorbed into the lacteals of the villi as chylomicrons before absorbing into the blood. Small intestine also provide immunity. Mucus layer prevent direct contact of the pathogen to the epithelium. Peyer's patches or aggregated lymphatic follicles neutralizes the invading pathogen or antigen. If you found my video helpful, then kindly subscribe, like and share my channel.